It's a great honor for me to receive the IFA Norman Borlaug Plant Nutrition Award. I'm delighted that my research over the last 30 years has contributed to several aspects of the new paradigm of responsible plant nutrition, including management of sulfur nutrition for crop yield and quality, improving human nutrition and health through biofortification of essential micronutrients, and reducing toxic metal uptake, and improving soil health through bioremediation. I started my research career doing crop sulfur nutrition research in the UK, at a time when atmospheric deposition of sulfur was declining very rapidly, and there was little information about crop sulfur nutrition. We developed a simple computer model combining sulfur deposition data, soil types, soil properties, and crop sulfur requirements to predict where sulfur deficiency was likely to occur. We established the effects of sulfur fertilizers on yield and quality of wheat, barley, canola, and legumes, and developed diagnostic methods for crop sulfur status. We also used stable sulfur isotope ratio and long-term experiments to quantify the cycling of sulfur in soil crop systems. This research forms the basis of sulfur fertilizer recommendation in the UK. Sulfur fertilizers are now routinely used in the UK and Europe. This research is estimated to produce £30 million benefit per annum to UK agriculture, according to the Homegrown Cereals Authority in the UK. Micronutrients are another topic of my research. Using Rothamsted long-term experiments, we found that the concentrations of essential micronutrients in wheat grains have decreased substantially since the 1960s after the introduction of high-yielding semi-dwarf wheat cultivars. This finding emphasizes the need to balance yield increase with nutritional value. We have conducted large-scale surveys of micronutrient status, especially selenium, in food crops and also in some human populations. Our data show that the intake of selenium is insufficient for large sections of the populations in the UK and China. We have established agronomic methods to biofortify cereal crops with selenium. Recently, we have also identified two genes that control selenium and sulfur uptake by rice. We are now in the process of integrating these genes into elite rice cultivars with the aim of enhancing selenium concentration in rice grain, which should benefit human nutrition. Agricultural soils in developing countries are under threat from contamination due to intensifying human activities. The most striking examples include the large-scale soil contamination in South and Southeast Asia caused by irrigation of arsenic-tainted groundwater, and cadmium contamination from mining and industrial activities. Accumulation of these contaminants in soil not only can cause yield losses, but their accumulation in food crops may also pose a significant risk to human health. Over the last 15 years, my research has focused on understanding how crop plants take up arsenic and cadmium from the soil, and what can we do to reduce their uptake. We found that the arsenic and cadmium are taken up through the uptake pathways for nutrients, arsenic through the silicon pathway, and cadmium through the manganese pathway. On the basis of this fundamental research, we have further developed practical solutions of using silicon and manganese fertilizers to suppress arsenic and cadmium uptake by rice crops. These research outcomes demonstrate that fundamental research in plant nutrition can benefit not only crop productivity, but also human health. Thank you very much for your attention.